Okay, moving along to our second talk in the Understanding Why ALS Occurs session. It's my pleasure to present our next speakers, who are a duo, a professor in the Department of Neurology and Neurosurgery, Dr. Stefano Stefani at the Montreal Neurological Institute at McGill University, and a postdoctoral fellow, Mathilde Chaineau, Dr. Mathilde Chaineau, also at the Montreal Neurological Institute, and working with Dr. Thomas Durkin as well, managing a iPSC platform in Quebec, which some of you may have heard earlier about induced pluripotent stem cells, and you're going to hear quite a bit more about what's happening in Canada for that. So without further ado, thank you for joining us, both of you, and the floor is yours.
Oh, hi, this is Stefano.
And this is Mathilde.
Can you hear both of us?
We can hear you and we can see your screen. Thank you.
Oh, excellent. So it's a great pleasure for us to be here. And it's really a pleasure to have this opportunity to share with whoever is listening to us. We don't know how, really some very, very exciting new developments, or recently new developments, I should say, but really exciting from the point of view, as you will see very quickly, of not only allowing us to address very important questions in ALS, but also, I think, importantly, emphasizing the concept of how significant it is and how important it is to engage. Oh, I can hear somebody's voice. Okay, to engage our patients into research, and I think this will be one of the main themes: patient engagement in research. So, how do I advance? Okay, so essentially, I think what we're going to do very quickly is to repeat something that I think most people already know quite well, that ALS is not only a disease related to the death of motor neurons. It's a disease that actually involves intensive communication, crosstalk between more than one cell types, not just neurons, but also neurons, as well as non-neuronal cells, glial cells, in the microenvironment. And this is a very nice cartoon taken from a previous review that shows that in a normal situation, motor neuron cells, shown here, are in communication with glial cells, such as astrocytes, resident immune cells of the nervous system, such as microglia. They make contacts with muscles at the neuromuscular junction, and there are other very important cells that play roles at the neuromuscular junction called the Schwann cells. So they are all communicating and talking to one another. And in the progression of the disease, it is believed that what happens is that this communication goes awry. For example, when the motor neurons start to exhibit the first signs of incorrect pathways or abnormal events, microglia can become, as it is usually said, activated. They can sense that something is not correct. Then as disease progresses and first symptoms appear, astrocytes also can become activated. And activation essentially means that they start to secrete a number of factors that can then have an impact and an effect on motor neurons. And as the disease progresses, motor neurons then undergo cell death, and it is believed that this is in part contributed by the communication with these non-neuronal cells, such as astrocytes and microglia. And so really what we are trying to do here at the MNI is to learn how to study this important communication and crosstalks between all of these different players using human patient-derived cells, particularly, and this is what we're going to talk about today, motor neurons, astrocytes, and microglia. And we're now in a process very soon of setting up protocols to derive also Schwann cells, and also Mathilde will tell you something about how we are already looking at the interaction, communication between motor neurons and muscle cells at the neuromuscular junction. So really that's what we're going to do today, give you a really quick a description of how these things can be done, how these human patient-derived cells can be obtained, and some of the applications that we are already implementing, and therefore some of the questions that can be studied. So what I want to show is something that many of you probably have seen before, what by many is referred to as the circle of hope. And the circle of hope, which can never be interrupted at any way, at any point, otherwise it wouldn't work, it really starts from the patient. The patient is the key point, the key starting point. The patient that decides and agrees to participate in research can become a source of biological samples as well as clinical information, which can then be stored in a fully accessible and open to everyone biobanking repository. Some of this material can be used to derive induced pluripotent stem cells from each individual patient. And then the beauty of all this is that from these individual induced pluripotent stem cells, we can then derive motor neurons as well as glial cells, for example, astrocytes and microglia, and then study them alone or in combinations in what many people refer to as modeling ALS in a dish, which means essentially modeling ALS in a laboratory using human cells, in order not only to conduct the discovery part, meaning to hopefully discover the pathways and the molecules that are essential for ALS pathogenesis or involved in ALS pathogenesis, but also to develop new and informative disease-relevant preclinical models that can then drive and direct in a much more focused ways the clinical trials that will then eventually lead to the identification of compounds, new compounds that will ultimately benefit the patients. And I want to emphasize that in this scheme, it is very, very important to achieve high patient participation in research. It is very, very important to be driven by strong scientific questions in order to take full advantage of the power that is offered by these new scientific approaches, and to then work together with industry towards the discovery of the compounds that will then eventually lead to new therapies. So now what we're going to do briefly is to show all of you what some of these motor neurons and astrocytes and microglia derived from human iPSC look like, and some of the things that we can do. And before I do that, there is a very important thing I want to say, which is that everything that we do at the MNI is under the philosophy and the auspices of open science. What this means that everything that happens along this circle of hope is publicly available. Nothing is protected or not made available. We share everything. We share biological samples. We share genetic and imaging data, obviously with all the necessary consents. We share all the cells that we can derive from iPSCs, and we work together with pharma, also under open science umbrella, in order to speed up the process of discovery. So Mathilde will now tell you something about motor neurons, and I will come back a few minutes later to tell you more about astrocytes and microglia.
So one of the first cell line we are generating at the platform, this is the motor neurons, because the motor neurons are the cells that are degenerating during the disease. So you can see here the timeline of the protocol we're using to derive motor neuron from iPS. So here it's a picture, bright field pictures of what iPS look like. So they grow as colony. And basically the idea is to use a combination of small molecule and growth factor to reproduce exactly what happened during the development. So we have different steps in our protocol. So we adapted the protocol that was previously published, and it's mainly the work of Carol here at the platform. And so during those different steps, there is one really important step that arrived after like 3 weeks, where we get what we call neuronal progenitor cells, and that's what they look like in the bright field pictures. And that stage is really important because we can expand the NPC and we can bank them. So meaning that every time we need to differentiate motor neurons for experiment, we don't have to go back every time to the iPS stage. We can start from the NPC. And so it's really important because it saves us a lot of time. So the other thing that I want to mention is that, of course, we have implemented a system of quality control here at the platform, where we want to make sure that at every stage we have the right cell, meaning that they express the right marker. So here is an example of immunostaining of iPS, and we are sure that they express nanog, and we have a wall panel of different markers that we use for every cell lines we're using. Also for NPC, so we are making sure that they expressed, for example, here SOX1, but also marker of proliferation, because those cells proliferate and we want to make sure that they stay like that. So then we differentiate the NPC into motoneurons, and that's a pictures of what the motoneurons look like after 4 weeks of differentiation, meaning 4 weeks after the NPC stage. And so we stain them for tubulin to see the entire network of neurons. But again, we make sure that they express the right specific marker for motoneurons, HB9 and ILATE1. And so those staining are being done for every cell lines we're using. Another way that we are recently implemented at the platform to characterize our line is that we can record the growth of the cells using a camera in an incubator that is directly connected to a computer. So we can make movies like that. And so what are you looking at now are some NPC that we have plated, and we have been started the differentiation, and we can look at them over time. So as you can see here, it's a highly dynamic process. We can follow them for weeks like that. So it's a highly dynamic process at the beginning with the motoneurons expanding their axon and neurites and like trying to connect, looking for each other. I think they're mainly looking for muscle, but in that case, we're just growing the motoneurons themselves. And it's a really cool thing to have, because by doing that with the control cells or with the cells that are derived from ALS patients, we can right away have an idea if there is something phenotypically happening in the patient cells. And so after 2 weeks, they start to form those clusters. So the cell body come together, and that's exactly what happened in the body with the cell body in the ventral horn, and then the axon going to connect with the muscle. So now Stefano is going to introduce you the astrocyte part.
Yes, so the concepts are similar. Again, I want to emphasize something that Mathilde also mentioned, that you have to really keep in mind that we can do this from virtually every iPSC line obtained from every patient that comes to our hospital. So this really is a very large scale program that we have in place. So astrocytes are also very important in ALS. I'm not going to go through the evidence for that, that they are. So let me show you briefly that essentially to make astrocytes from human iPSCs, we have 3 main steps. We first have to make neuroprogenitor cells, like Mathilde said. Then we have to force these neuroprogenitor cells to choose to become glial cells. This is what we call glial commitment. And then we have to tell them to make specifically astrocytes. In vitro, we can do that in about 1 month. And this is the work done mainly by a research associate that works in my lab, but also works with Tom Durkin and a lot of Mathilde as well. And this is how we can examine at every single stage the property of the cells. We can make sure that we have nice, robust derivation of neuroprogenitor cells. For example, only after 2 weeks. We can monitor the formation of the very first committed gliogenic neuroprogenitor cells that will express a particular protein called GFAP. And then we can look at about 1 month at the formation of astrocytes that will express a number of different astrocytic markers. And what we know is that on average, from culture to culture to culture, about 70% of the induced cells express, for example, this particular astrocytic marker here called S100 beta. And the same is true for other markers. So the point I want to make is we can get robust derivation. Importantly, if these cells are astrocytes, they also have to look like astrocytes in terms of what they do. And what astrocytes are very well known to do is that they can communicate with one another through this form of intercellular communication, which is mediated by wave-like propagation of calcium currents. So one of the things that Vincent can do is to measure the ability of these induced human astrocytes to exhibit this kind of wave-like propagation of calcium currents. I'm going to show you very quickly a movie here that shows the key color to look at is the green. That will show to you this. We started here, maybe, no? Okay, you will see how the cells are having this non-synchronous calcium waves that you can see here, which is usually indicative of the ability of the astrocytes to talk to one another. So we can stop the movie, and essentially here is that the calcium waves are used as an indicator of the physiological calcium dynamics. in these induced astrocytes as a sign of the fact that they in fact are behaving as they're supposed to. And lastly, I want to tell you something very exciting about microglia. This is very, very recent work done in my lab by Yeman Tang, and now other people here are also successfully working on the derivation of microglia. Here, the process is different. We need to use the human iPSC to first make so-called primitive streak-like cells, which are the very, very initial precursors of the myeloid lineage. Here we are made cells of the myeloid lineage, and then we have to drive them to make specifically microglia, or cells that look like microglia, and we can follow this through the expression of very, very specific markers. So for example, after about 36 days in culture, so again about a month, we can see that almost all of the cells that are cultured express this particular microglia progenitor marker called RANKS1. And then about 10 days later or so, we see that more than 90% of the induced cells express this particular microglia marker called P2RY12. So very, very robust generation of microglia-like cells under these conditions. And very quickly, I want to tell you why they really look like microglia. I will skip this one. Well, one of the things the microglia do is that they eat the dying cells, so they can eat cells that are undergoing death for a number of reasons. This process is called phagocytosis. And this picture shows that, in fact, many of the induced cells have phagocytosed these red fluorescent beads that you now see inside the body of the cells, indicating that, in fact, they have the capacity to phagocytose. And so this is an indicator of physiological properties of these induced cells. And lastly, like astrocytes, microglia also have wave-like propagation of calcium increases. So Vince also looked at this property in microglia. These are now cultured human microglia. And again, you can see how the cells are non-synchronously exhibiting calcium waves, which are suggestive of physiological properties of these cells. So now Mathilde will tell you quickly some of the very exciting things that can be done with these cells once they are derived.
So now that we have all our cells, what are we doing at the platform with them? So our goal is to develop some assay that will model the disease and then do some drug screening to test compounds and small molecules that can revert the phenotype observed in the disease. So at the beginning, we were thinking that very naively that the motor neurons derived from patients will show a decreased survival without having to stimulate their death in comparison with control. And that's not what we've been looking. The neurons were growing normally, were behaving exactly as the control neurons. And but the thing is that it has been also reported by other groups. So in order to decrease the viability of the cells, we need to induce oxidative stress with sodium arsenate or excitotoxicity with glutamate. And as you can see here, in control cells or in the SOD1 line, we decrease the viability of those cells using those stimuli. So, and interestingly, those stimuli are used to decrease viability, can be mediated by other cell types in the organism, like the astrocytes. So we decided to go a little bit further and to develop co-culture systems to be more close to what happened physiologically. So very quickly, this is an example of the co-culture we're developing here, and it's co-culture between astrocyte and motor neurons. So you can see a bed of astrocytes, and on top of this, we add the motor neurons and differentiate them. So this is a picture of control astrocyte cultured with control motor neurons. So everyone is happy, everything is going well. The idea would be to see if we use astrocyte from patients, are they gonna secrete some factors that are gonna trigger the death of the motor neurons. And very, very quickly, another thing that I would like to mention, it's because it's an important part of what we're doing here, is we really want to model the neuromuscular junction in a dish. And to do that, we used iPS-derived motor neurons, and we are also using myoblasts that we derived into muscle. So for now, we're doing that with human muscle, but like isolated from primary like biopsies. But we are working on developing a protocol that will allow us to get muscle from iPS, so that we will really be able to get the neuromuscular junction of healthy individual versus patient and see what's going on and what's the difference. And eventually to use a model for drug screening as well. So in conclusion, we have implemented at the platform in the lab of Dr. Stefani a robust way to generate functional iPS-derived cells like motor neurons, astrocytes, and microglia that are implicated in ALS disease. And really, the ability to generate different cell types from iPS from the same patient is really important because it allows us to reproduce a more physiological system that will be used to screen compounds. We have been developing some co-culture model to study the non-cell autonomous mechanism responsible for the loss of motor neurons observed in the disease, like astrocytes and soon microglia. And finally, we are implementing a system where we can reproduce a neuromuscular junction from healthy individual and ALS patients. And really, ultimately, our goal is to develop high throughput compatible drug screening assay for ALS. So the idea would be to scale up the co-culture assay that we're doing to do some drug screening, and we are also implementing all the automation here at the platform in collaboration with NeuroCDRD to make that faster. So with that, we'd like to thank Vincent and Yeman from Dr. Stefani's lab for their help with the project, and also the people from the iPS CRISPR platform led by Thomas Dorcan and Ted Fon, Maria, Mark, Vincent, Gilles, Carol, and Wolfgang that are involved in the projects. As well, we'd like to thank the funding and our partners, ALS Canada, Hudson Grant, SGC, and Takeda. And thank you very much for your attention.
Thank you both for that. That was really fantastic, and it's come such a long way. So really appreciate you presenting it to us today. One quick question. So you talk about the co-cultures that you're starting to develop, and wondering if there's a drive eventually or a possibility to be able to eventually take some of these varying cell types and reform sort of a human 3D model in a dish, whether it's a 3D culture system with varying cell types or even an organoid type such situation. Is that possible, you think, in the future?
Yes, excellent question, David. And we've been really working very hard on this. So at the MNI, we have already successfully developed protocols to grow brain organoids. And now we have begun to work on the generation of spinal organoids. So the technology exists. There are very few people around the globe that do it, but we are in touch with them. And you don't develop as nicely structured 3D mini brain as in the case of, as I said, the brain, but now also for the spinal cord, this is coming along. So I am very optimistic that this is coming and that we will be there quite soon.
That's so cool. Well, thank you both for your time, and we really appreciate it, and look forward to seeing how it all develops in the future. So have a great day.
Thank you so much.
Thank you.
Yeah.